All right, everyone. I went ahead and popped a few slides back really quickly because there was one point of clarification I wanted to make based on a few questions I received either during office hours or via email. One thing I want to note is that the DNTPs, the deoxyribonucleoside triphosphates, they serve two important roles. It bears worth repeating, and it's also good to note that if I'm repeating something that's in blue, it's important to know. This is the source of the new and incoming nucleotides during replication. So if you actually look, you can tell at this DNTP on the bottom left, that is actually a thymine with just two extra phosphate groups on it. And so those two phosphate groups are actually removed, and you can see that here that the DNA polymerase helps remove them, and they're used as an energy source during replication. Okay, so these DNTPs are actually the nucleotides themselves, and that the two extra phosphate groups on them serve as a source of energy during the replication here. So now, yes, go ahead. Oh yeah, sorry. It is functionally the same thing. So there's like the ribose that's part of like what makes up a DNA nucleotide. So it's functionally the same thing. Yeah. Thank you for asking about that. Are there any other questions on this slide about DNTPs? Or I'd be open to answer any questions about anything we talked about from last lecture that needed clarification. One thing that always bears worth repeating is that if there's a burning question that you have, that's something that you're wondering about, it's likely that someone else might have that same question. And if there's something that's not clear, that's on me, not on you guys. So, yes.
Another question about the piece, or yeah, the piece. It says like nucleoside, but I feel like in one of the pre-lectures it said that the nucleosides were like the units of the DNA without the phosphate groups, and like the nucleotide like with one phosphate. So I was wondering why.
That you're saying with the two extra phosphate groups, that's when the nomenclature that it uses the term nucleotide as opposed to that. Yeah, so that okay, thank you. So typically it's a nucleotide when it has the one single phosphate group on it. Does that help clarify?
Sure. So the margin of
as to my understanding, we typically use that when it has the extra phosphate groups on it. That's always been my understanding. Thank you for asking that. I appreciate that. All right, so I'll go ahead and jump back to where we were before. We were at this slide here. We just finished talking about the leading strand versus the lagging strand, but it's worth repeating again that at each replication fork, and again, this replication fork is growing, will be melted further, and the fork will grow to the left here. The leading strand is the new strand of DNA that is being polymerized or growing into or towards the replication fork. As the fork opens up more and more, the opposite strand, the lagging strand, there will be more available DNA to be used as a template on the top strand. And that's how we get these fragments, these Okazaki fragments. And it's always important to note that which is the leading strand versus the lagging strand is always relative to which replication fork we're at. Because this fork is part of one single replication bubble. If we look at the other end, at the other replication fork, what is the lagging strand here will be the leading strand there because it's growing towards that replication fork. So it's always when we're talking about the leading strand versus the lagging strand, it's always relative to which replication fork we're at. So one question that I'm sure a lot of you had was when we talked about the role of primase, how it lays down this RNA primer, you might have been thinking to yourself, and it would have been okay to ask this at the time, why do we have RNA in our DNA? That doesn't seem right. How does that get addressed or tackled? We actually have one DNA polymerase, another DNA polymerase species that helps tackle that, DNA polymerase 1 in prokaryotes or DNA polymerase alpha in eukaryotes. So from here, I'll call it either DNA pol 1 or DNA pol alpha. It has the same functions as DNA pol 3 or DNA pol delta. It can serve as a polymerase in the 5' to 3' direction. It can serve as an exonuclease, the proofreading function, in the 3' to 5'. But it can also serve as an exonuclease in the 5' to 3' direction. Why that's important, again, an exonuclease removes nucleotides at the ends here. What it'll end up doing is that it's going to be the polymerase that is responsible for removing our RNA primer. And as it does so, it's able to also function as a polymerase, where it removes the RNA primer and helps promote the growth of the growing strain with actual DNA now in that site. So what that looks like, and a key thing to note is that's actually slower than DNA pol 3, so its typically main function is to try and help remove these primers. So what that looks like is we have our template strand on the top. Primase has come in, this DNA is melted and separated. Primase has come in and put down an RNA primer. DNA pol 3 or DNA pol delta is going to see that. And again, this is important to note here. Our DNA polymerases cannot initiate and create a new DNA, cannot initiate DNA polymerization without something there. It needs this primer to grow off of. And so DNA pol 3 or DNA pol delta will see that and produce the new growing strand that is anti-parallel and complementary to the template strand. And then on the other side of this primer, there's going to be, again, in eukaryotes, there's multiple origins of replication. So there's going to be replication happening along multiple sites of a linear entire chromosome, entire segment of DNA. And so there's going to be DNA that's going to be replicated that abuts to this RNA primer. Again, we can't just leave RNA within our DNA. This needs to be removed. So this is where DNA pol 1 or DNA pol alpha comes into action. It'll come in and it'll both remove and lay down new DNA that is anti-parallel and complementary to the template strand here. Once that's laid down, as well as with these Okazaki fragments, I think there was a question in class where someone asked, we have these Okazaki fragments, but they're fragments, they're not joined together. Sorry about that. It's always a great look when you have alerts like that pop up. We talked about, someone asked about the Okazaki fragments, about how they're disjointed and how is that overcome, as well as when we have the RNA primer replaced with DNA by DNA pol 1 or DNA pol alpha. We need to join together those fragments or those segments of DNA. So what's really important is that there's going to be a key enzyme that's responsible for that ligation. Ligation is the joining together of those fragments. And so the top half here is meant to depict DNA polymerase 1 removing the RNA primer in red and replacing it with DNA. And then what's really key to note here is that the name of the enzyme or class of enzymes that are responsible for ligation, joining them together, is called DNA ligase. DNA ligases ligate together, join together DNA fragments so that we have a single contiguous double-stranded DNA that makes up that long segment of that chromosome. And it's meant to fill in those gaps to make sure that there's no gaps at all within our DNA during replication. So putting all of this together into one cohesive image for how DNA replication happens, we have our initiator proteins. And again, if a term's in blue, it's important to know. We have our initiator proteins whose job it is to identify the origins of replication. In prokaryotes, there's usually a single one, that consensus sequence. In eukaryotes, there are multiple along a single chromosome. With prokaryotes, that initiator protein can actually separate. With eukaryotes, it can't. So we need our helicases. Our helicases are professionally tasked with separating or melting our DNA strands at the site of the origin of replication. And again, we have our single-stranded, this isn't listed here, but our single-stranded binding proteins that help stabilize the melted DNA, because DNA, it's energetically favorable for them to rejoin and anneal back together. When they separate, we get that supercoiling that happens at the ends. And so to alleviate that, gyrases will help, will come in to, specifically, again, type 2 topoisomerase is the gyrase here that we've talked about. It'll help relieve that supercoiling. To initiate actually initiate replication here, DNA polymerases again cannot lay down new nucleotides or create a new growing strand unless there's nucleotides already there. So that's why we need the RNA primer. Primase's function is to lay down that RNA primer. Once that RNA primer is present, now DNA Pol 3 or DNA Pol delta can extend that strand off of the RNA primer. And to help replace that RNA primer, DNA Pol 1 or DNA Pol alpha will do that and replace the RNA primer with DNA. And finally, there's going to be lots of fragments or disjointed segments. And it's DNA ligase's function to help join together at the phosphate backbone is where our DNA strands are joined together. So it creates a phosphodiester bond between the phosphate backbones of adjacent nucleotides to help ensure that we have no gaps in our DNA and it's contiguous. I'm going to stop here and wait for a couple questions. Yeah.
So you might have already answered, but how does the DNA polymerase start? Like get rid of that what time?
Yeah, so I'll go back. Actually, like this slide. So at this point, so it's important to remember that DNA polymerase can only work. I'll try and stay close to the microphone, forgive me. The question is, just to make sure I reiterate the question for the recording. Your question was how does DNA polymerase 1 know to remove there, or all DNA polymerases need already laid down nucleotides to work off of. So in the case of DNA polymerase 3, those nucleotides are going to be this RNA primer. For DNA Pol 1, it's going to be these nucleotides to the right that'll enable it to extend off of that and remove this RNA primer at the same time. Does that help clarify?
So that one prime there. That's a couple.
It initiates, it's the initiating event for DNA Pol 3. For DNA Pol 1, it's not that this is, again, where everything's growing this way. So this is 5' to 3'. These nucleotides are the priming nucleotides for DNA Pol 1 to remove the primer in the 5' to 3' direction. Got it? Okay. Yes. How does, you said how does ligase? Yeah, so ligase, it's not reading the DNA at the nucleotide level. What it's doing, so it's ligase's job is to join together fragments of DNA at the single stranded level. So what it's looking for is gaps in the phosphate backbone of our DNA and helping to create phosphodiester bonds. And those are bonds between phosphate groups that help fill those gaps. Yeah, it looks for gaps in our DNA backbone and works to fill those gaps. Thank you for that question. Yes. Multiple ligases. This is happening all across the DNA. Yeah. You're asking, should there be two DNA ligases for one replication bubble? I would actually, now you're asking a really good question. The question is, are there only two DNA ligases for each replication bubble? I actually don't know. The reason why I'm saying it that way is because at the heart of your question, it seems like you're asking, do DNA ligases function at multiple fragments simultaneously, or does a single DNA ligase work down across broken phosphate backbone and working in that direction? Actually, I don't know the answer to that question, but I will get back and I'll try and share that next class. Thanks for asking that. Yes.
This is kind of similar, but with the primates instead, does it take several primers with a ligand strand?
Yes. Yeah. All right, we're going to go ahead and press on. So we've put it all together here. There's one important problem that needs to be addressed for this for replication. So an important thing to note is that, again, our DNA is packaged up. So here we've taken, you can envision that this is one entire chromosome that's been laid out linearly. As we go through replication, if you can envision having primase laying down an RNA primer all the way at the end here, we're going to have eventually the entire linear DNA that's going to be replicated. Once the primer has been removed, and this gets back to the question you were asking earlier, once the primer gets removed, there are no nucleotides upstream for which DNA pol 1 can now lay down and copy this end of the DNA. This is called the end replication problem, because what's going to happen now is that end of the DNA will not be replicated in the subsequent daughter strand. And so it's going to be shortened by that much, by the amount of an RNA primer. Now, when this daughter strand is going to undergo replication, the end replication problem will happen again. There's going to be an RNA primer at the end. When it's removed, there are no nucleotides upstream from which DNA polymerase can grow off of. So now the subsequent strand replicated here is going to be shorter by that much more. These ends, you might have heard of this term before, but these ends of our chromosomes are called telomeres. There's the problem of these telomeres shortening over the course of replication that needs to be addressed and taken care of. We have a class of enzymes that help repair our shortened telomeres to ensure that this end replication problem doesn't persist. So we have this enzyme that's called telomerase. It is an RNA-dependent DNA polymerase. And so in that name, you might hear it's RNA-dependent. Well, we're working with DNA, so where's the RNA coming from? What's really fascinating is that telomerase provides its own primer here. Packaged within telomerase is this segment. You can see it here in this first image of telomerase here, that there is this RNA primer right there from which it can use to elongate and restore the telomeres at the end of our chromosomes. What's really important about this is that as we age, some of you may have already heard this before, as we age, our telomerases lose their function. We lose telomerase, and one effect of aging on our DNA is the shortening of these telomeres. Now, these telomeres, this is an evolutionary advantage where our telomeres are not encoding for our actual genes here. But what ends up happening over time is that as our telomeres shorten, you can think of it as almost like a biological clock, whereas telomeres shorten, we're gonna reach a point where actual encoding regions of our DNA will now be affected and shortened. So the erosion or the shortening of these telomeres is an event that can lead to the development of senescence. Senescence is this process of cells undergoing cell cycle arrest, and it's this age-associated phenomenon where cells undergo this really severe dysfunction. And a lot of research, including a lot of Mackenzie's work for a dissertation, is tied to senescence and demonstrating that it lies at the heart of a lot of disorders and diseases. So if you want to talk about senescence, I would definitely direct you towards Mackenzie and peppering her with your questions. Or not. So taking us all the way back to anthrax now. So we talked about how anthrax, when it's in unfavorable conditions, it'll form these spores. And so when, particularly with inhalation anthrax, these spores will be inhaled. Macrophages, or remember, there are these key phagocytes within our innate immune system. They will phagocytose these spores. What ends up happening now is that these spores are now in a more favorable condition. And what's going to end up happening is they're going to germinate and proliferate massively to an extent where they're going to eventually lead to cytolysis, or the lysing of macrophages. You can think of lysis as just the blowing up of a cell. And so it's going to destroy the macrophage. And once that happens, sorry again, once the macrophage undergoes cytolysis, this bacteria will be able to cross the epithelium. What's really important here is that within the lung, crossing the epithelium gives it access to our bloodstream. And again, we talked earlier about sepsis, this dynamic of when a local infection goes systemic. It goes systemic once it gets into the bloodstream. So when the spore germinates, proliferates, destroys macrophages, and crosses the epithelium in the lung, once you cross the epithelium, you have readily available access to the capillaries. And these capillaries in the lung, there's a lot of diffusion and transport across the epithelium within the lung. And so once it can get there, that's where sepsis can take place here. There are what's important for us to talk about before we address how we can tackle anthrax infection is defining some key terms. And so antimicrobials is an entire class of drugs that can target bacteria, virus, fungal infection. So it's a whole host, a whole class of drugs that target each of those. Antibiotics specifically target bacteria, and usually they're going to be synthesized by other microorganisms that helps them fight against other ones. What's key to note here is that usually with antimicrobials, the broader class can include both natural or synthetic substances. So there's a lot of times where these antimicrobials are identified in some sort of either a naturally occurring organism or within some unique specific plant. Or once we do a screening and identify them, they can then be synthetically created off of that. And so what's key to note here is that antimicrobials can usually include both natural and synthetic substances. Antibiotics are typically naturally derived, but then can oftentimes be used as a direction to kind of create the antibiotic drugs from what is already naturally occurring. For our antimicrobials, there's what's important to note are some important properties for each of them. As you can imagine, it would be really important for it to be selectively toxic, meaning that we'd want it to be toxic to the pathogen, to the microbe, but not to the host. So we'd call that selective toxicity. You'd also want it to be stable. You don't want it to have, you don't want to create a drug that would require really unique storage conditions where it would be hard to logistically hard to deal with that. You want it to be inexpensive, and you want it to be really easy to administer. In addition to that, this next point is going to be really important. You want it to be bactericidal, not bacteriostatic. Meaning, if you have 100 spores in one tube, and 100 spore in tube 1, and 100 spores in tube 2, for example, you'd want it to not just stop the growth of these spores in these tubes, because typically, if you have favorable growth conditions, they'll grow, and 100 spores will turn into 100,000. But you don't want bactericidal would go from 100 spores to 0. Bacteriostatic would go from 100 spores and stay at 100 spores. So bacteriostatic means it stops and inhibits the growth of the bacterium. Bactericidal means it kills all of them. So you want to make sure that whatever this drug is bactericidal. And you want to make sure, and we'll talk more about this in a moment, that it whatever the drug is, it's not something that can be these pathogens or these microbes can become easily resistant to. Depending on the infection, where it takes place, typically you want to be able to have appropriate tissue distribution. As is the case with a lot of antibiotics or antimicrobials, there's oftentimes allergic reactions, and so you want to be mindful of that. And you want to have the appropriate spectrum of activity, meaning that it's able to be bactericidal to cover the entire lifespan, or not the entire lifespan, rather a better way to put it is to enable that it has sufficient time to be active and ensure that it destroys all of the bacterium there within this infection. We have two, and this might be something that you all have heard before. We have generally a couple of classes of antibiotics. We have broad spectrum versus narrow spectrum. Broad spectrum antibiotics are typically drugs that will go in and try and kill any antibiotic or any bacterium. Narrow spectrum meaning that it will focus on and try and target specific subsets of bacteria. So, for example, we talked beforehand about gram-negative versus gram-positive bacteria. So, one level of narrow spectrum antibiotics would be those that target either gram-negative or gram-positive. There's multiple levels at which they can focus in on, but that's one way in which you can differentiate what antibiotic you'd want to use. The problem, the thought might occur that broad spectrum antibiotics might just be a safe approach where you can just destroy all the bacteria and ensure that you're getting rid of this infection, particularly if you're dealing with something like anthrax. The problem is that there's two problems, I should say. One, we have healthy probiotics, healthy bacteria that we need. It's always funny to say this out loud because I kind of forget this each time. We have about pounds of healthy bacteria within our body that we need. So destroying them can be highly problematic for various organ systems. Additionally, when you use a broad spectrum antibiotic that wipes out all of the bacteria, what you're doing is creating an environment that is suitable for resistant bacteria to take root and germinate and grow. So, for example, there's an analogy that might be helpful is trying to think about how if you wipe out all of the predators within an ecosystem, you're creating new problems that arise and emerge. So, for example, within an ecosystem, if you wipe out all the predators, what's going to end up happening is that the prey for which they usually help regulate their population, they're going to decimate the ecosystem and consume lots of the foliage that creates multiple problems throughout it. And so it's a loose analogy, but the main take-home point here is that broad spectrum antibiotics can really destroy your healthy bacteria that you need, and will also create an environment that is suitable for antibiotic resistant bacteria to take place, germinate, and grow. And so these are the key reasons why we typically try and avoid using broad spectrum antibiotics right away. So the antibiotics that we typically use will function at multiple levels. They might, and back during our immunology lectures, there are points at which I tried to have like a virtual asterisk that I identified what's different between our biology or our microbiology, our cell physiology versus the physiology of microorganisms or pathogens. The way antibiotics are designed also kind of function the same way, meaning that some antibiotics, such as penicillin, target the cell wall. Our cells don't have a cell wall. So this is one way in which it can have selective toxicity specifically to bacteria. Another is looking at cell membrane and function and parts of the cell membrane that are unique to fungi that our cells don't have. And so an example of that is meconazole, that is able to target unique features of the cell membrane in fungi that are not present in our own cell membranes. There are specific antibiotics that target the machinery of protein synthesis that is unique to bacterium. We have streptomycin, erythromycin, and tetracycline. Key metabolic enzymes that are unique to bacteria that can be targeted, sulfonamides. And then finally, nucleic acid inhibitors that can inhibit replication. We're going to focus on these. Look at and target transcription as well. So the drug that we briefly touched on at the beginning of this replication series was on Cipro or ciprofloxacin. It's widely considered to be one of the most powerful antimicrobials out there. It is typically only effective for inhalation anthrax. This is one point that I'm still trying to look into, but this is one thing that's been made clear based on how it's administered. And you can tell that its structure is pretty unique, and that's something that's not usually naturally occurring in our human physiology. And so it falls under the category of fluoroquinolones, and it specifically inhibits bacterial DNA gyrase. Okay, based on what we know about gyrases, what do you suspect Cipro is going to do to anthrax bacteria? Good. Yes. So you did get the hint that it inhibits replication, but based on it targeting DNA gyrase, how do you suspect that it's inhibiting replication? Go ahead. Exactly right. What ends up happening is that because we're inhibiting DNA gyrase, this supercoiling occurs outside of this replication bubble. And because of that, the DNA cannot be separated or melted any further. As a result of that, this is going to block and inhibit DNA replication within these within the anthrax bacterium. So I'll actually take a moment and stop here for questions. Yeah, I can stop for a couple questions here. Yes. DNA polymerase 1 can replace, it is able to replace the RNA primer with DNA as long as it's not at the end. If it's at the very end, there's no DNA available upstream from it to elongate off of. The key thing to note with the end replication problem, like what's at the heart of it, is that DNA polymerase always needs nucleotides to grow off of. So at the end of our linear DNA, once you're at the end and the RNA primer is removed, there's no DNA for it to elongate off of upstream of it. Does that help clarify? Yeah. Yes. Mm-hmm. Mm-hmm. Yeah, that's a great question. So it's providing it is oftentimes providing telomerase will oftentimes provide the primer for it to help grow off of what's at the what's on the template strand. But at the so it's it is, what's the right way to say this? Yeah, it's doing normal polymerization of serving as normal polymerase going off of the template strand. But in your question is something important, that these telomeres do not encode for any genes. They are functionally a biological clock. You can typically tell how old a cell is by looking at its telomeres. You can also tell how old a cell is. You can also tell how old a cell is by how active telomerase is. What's important to note is that for those of you who work in a research lab or have been in a research lab, we typically keep our cells in a liquid nitrogen tank. And some of those tanks, you might see some cells that have been around since before I was born, since like 1985. Those are because, typically our telomerase as we age becomes less active. So it's sad for me to say my telomerase is less active than y'all's telomerase. There are cells where their telomerase is engineered to always be active. We'd call those immortalized cells. So for an example, there's a story of Henrietta Lacks where her cells were taken and used and studied extensively in biomedical research. And so like this is one example of fairly unethical behavior where there was something unique identified about her telomerase, where her cells can now grow pretty rapidly. And so it is most likely that either in MR5 or MR6 and a lot of liquid nitrogen tanks, you'll find what's called HeLa cells, Henrietta Lacks cells, because of her unique telomerase in her tumor cells. But again, we typically will immortalize these cells by engineering their telomerase to always be active, so they're always replicating and will never stop growing over time. I went down a rabbit hole, forgive me, but I also thought it might be beneficial and helpful for y'all to hear that. We have about 8 minutes, so I'm going to press on because I think we have a few important things for us to discuss. So a few key things about mustard gas and its role and how it functions or how it relates to replication. The way in which it acts as a vesicant, as a blistering agent, is not entirely known. It's thought that the basal keratocytes that are particularly vulnerable to this blistering causes an outswelling of the skin. But what's key to note is that mustard gas's main deleterious effects are on our DNA, primarily by creating a modification on our guanines, on those specific nucleotides within our DNA. We talked about and we're quizzed on how mustard gas acts. It's an alkylating agent. And so what it's doing is that it's providing an alkyl group here that will serve as a bridge between guanines. What it's going to do is that it's going to end up cross-linking them together. It's going to create this bridge that's going to affect replication. What that looks like is there can be intrastrand cross-links between adjacent guanines here. And so this can be a bit of a problem where it'll affect the ability of polymerase to go down and grow a new strand that is being created during replication when it runs into these intra-strand crosslinks between guanines as a result of mustard gas and the alkyl group it adds. There can be a free hanging alkyl group here off of a guanine that has no adjacent guanines or near guanines around it. What is particularly deleterious here from mustard gas is when it creates these interstrand crosslinks between these guanines, whereby at this point, the DNA cannot be melted. It cannot overcome this cross-linking. And so it actually affects the ability for helicase to separate our DNA there. And so what's going to end up happening is that polymerase is just going to run into this and helicase cannot separate the DNA there. And so this is going to stall replication and it's going to create something that's called a strand break. There's a couple of different kinds of strand breaks. We'll talk about them in a second. Some are more harmful than others. What is important to note is that various forms of mustard gas have been considered to be used as a chemotherapeutic agent to fight and target cancer cells. Again, because if it's affecting replication and tumor cells typically grow massively and rapidly, this might help, this might be one way to fight and target tumor growth. The consequence, the issue with that is, it's hard with any chemotherapeutic drug or with any drug therapy, cancer therapy, forgive me, it's always important for it to be targeted. And so for this, if you're affecting replication, that's a really key function for our normal healthy cells. And so that's one of the reasons why this is still at the testing phase, for lack of a better phrase. So I referred to DNA breaks. There are breaks that can happen. Before we talk about breaks, there's also effects that can happen on specific nucleotides, like we talked about with mustard gas. What can happen is that there can be a depurination. What ends up happening is that within this nucleotide, we can lose the purine group here, the purine sugar, and we can go from guanine to this depurinated sugar here. Another naturally occurring form of damage to a nucleotide base is a deamination event, where we lose an amine group here, whereby a cytosine will be altered and actually become one of the nucleotides that's unique to RNA. It'll become uracil. Again, big picture take-home message here is that there are naturally occurring things that can alter or change our nucleotides that can be problematic. And so there's a depurination event. We lose the purine sugar here. There's a deamination event where we lose the amine group here. And in this case, cytosine can become uracil. There's also the formation of thymine dimers, where adjacent thymines via UV light or UV radiation can actually become cross-linked together as well. Yes. Yes, these are just naturally occurring damages to our nucleotide bases that happen naturally. To dovetail off of that, we have mechanisms in place that help repair anytime there's a damage to a specific base. So we talked about how our DNA polymerases have an exonuclease activity, how it acts on the end of a DNA fragment. Endonucleases are a function in the middle, right in the middle of a DNA fragment or a segment of our DNA. So to help address these different forms of base damage that can happen naturally, we have repair endonucleases, which means that, for example, in this illustration here, this jagged orange line, forgive me if the color's wrong, represents damaged DNA base or damaged DNA base pairs. And what happens is that the repair endonuclease is going to help identify that wrong base there. It's going to remove it. And then DNA polymerase, because there is DNA or nucleotides available from which it can extend off of, will grow in the 5' to 3' direction. And DNA ligase will help ligate together or piece together the gaps in the phosphate backbone of the DNA here. So to link the previous slide with this slide, there are naturally occurring forms of base damage repair. And there are mechanisms like repair endonucleases that help fix those damaged base pairs. Really harmful forms of DNA damage are going to be actually double-stranded breaks. Yes. Yeah.
Is there is it more likely DNA fall through?
This goes off of something that I actually think I glossed over really quickly. It is most likely to be DNA Pol 3 because DNA Pol 1 acts a little slower than Pol 3. But because of DNA Pol 1's unique function as a 5' to 3' exonuclease, it's uniquely tasked with removing the RNA primer. But as a 5' to 3' polymerase, DNA Pol 3 is going to outperform DNA Pol 1. Thank you for asking that question. I'm going to do my best to finish this slide. It'll be 1 minute long, so forgive me. Double-stranded breaks of DNA are particularly problematic, and there needs to be an endogenous mechanism. There needs to be a way in which our cells can help repair and address these double-stranded breaks because they are so problematic. So there's going to be two mechanisms by which this can happen. We have non-homologous end joining, which is largely the addition of random pairs of DNA that are meant to help piece together this double-stranded break at the overhangs to make sure that that it sufficiently matches up. And what can happen as a result of this, so the non-homologous, this implies that there's going to be some differences that are going to be created because of how this mechanism takes place, because of the random addition of nucleotides. The other mechanism is homologous recombination. What's really unique here is that if we have a double-stranded break and we have these overhangs depicted at the section that's called end resection, what's really unique is that homologous recombination. It's called homologous because if you remember, for every gene, we have two copies. For every chromosome, we have two copies of our chromosomes that we have. Homologous recombination leverages the fact that we have two copies of these genes, and it'll actually use the other chromosome as a template here to repair what is supposed to be here at these segments of our DNA. And so the key thing to know with homologous recombination is that, again, it's using the other copy, the other chromosome, the other copy on our other chromosome as a template here to create the strand that is meant to be there, as opposed to random addition of nucleotides, and it'll be annealed and ligated back together. I'm going to go ahead and stop here. So I'm going to say sorry for going over. I'm going to post a recording of the remaining slides and post that on Canvas. It's only going to be 2 slides. So it'll be a short video. I'll actually, you know what, forgive me. I'm just going to hit this one slide and this is the last one. Homologous recombination is leveraged in a few different mechanisms. One is CRISPR-Cas9 genetic engineering. A lot of you talked about that in your projects. What's key to note here is that it's one of the biggest discoveries in science in a while. It usually takes a long time for someone to be awarded the Nobel Prize, on the order of decades. This is on the order of 5 years that they were awarded the Nobel. And so CRISPR stands for clustered, regularly interspaced, short palindromic repeats. Cas9 is an endonuclease. So CRISPR typically, it's this like endogenous mechanism that was identified as part of the innate immunity of microbes. But what ends up happening is that it's actually used as a template here and uses Cas9 as an endonuclease. There's a few different endonucleases that have been identified that function better than Cas9. I'm gonna stay close to the recording, sorry. And so largely what happens is that there's a double-stranded break. Homologous recombination then is leveraged using this guide RNA that's loaded into the endonuclease and used as a template to be able to rewrite our DNA during recombination here. If any of this fascinates you, we have a couple of folks studying DNA replication. Sorry for going 3 minutes over. Thank you for bearing with me. Have a great day.